Hello, everyone. Thank you for the invitation and the possibility to give this talk. And it's really nice to be here in Oxford in an actual live meeting. So my lab in Jyväskylä, Finland, we usually study phage bacterium coevolution and ecology. Mainly, we have been working with flow bacteria. But I was thinking, since we have had this kind of very long-term kind of side project, which has now evolved into an actual project 1.5 years ago, I would present you what we've done with the single-stranded DNA phages, the flip phage, which I will tell a little bit in more detail. And so here you can see a nice picture of our campus. So we live in the central Finland. So during the winter time, the lake is frozen, and in the summertime, the water is flowing freely and nice and warm. So we have quite a bit of large seasonal differences, which is one of the key players here in the ecology of phage bacterium interactions in that natural lake environment. But first, I will introduce you our pet phage, Flip. And flip means oops, this was a wrong button. Let's press that one here. So flip is an abbreviation of Flavobacterium infecting lipid-containing single-stranded DNA phage. So it's a unique phage completely with all these different characteristics of having a non-tailed capsid, icosahedral capsid, with an internal lipid membrane and a single-stranded DNA inside. So within the capsid, the genome is approximately 9,000 nucleotides long. It's a circular DNA molecule, and it encodes for 16 coding sequences. 5 of those have been identified to be structural proteins, marked here in this figure in a lighter blue color. There's not much sequence similarity to other known phages, but we've done the structural identification based on sequencing some of the structural proteins from kind of the N-terminal sequencing. There is some sequence similarities in P14 to putative transglucosylases, so it has lytic enzymes, and also to a rolling circle replication protein. But this phage does not encode any DNA or RNA polymerases or ATPases. So although we don't have much sequence similarity to other known phages, when we look at the structural level, the fold of the major capsid protein, the P8, we can, again, wrong button, sorry. We can see the structural similarities to, for example, PM2, the corticoviridae phage major capsid protein. So the major capsid protein is a double beta barrel. Has a double beta barrel fold. And this protein fold is found also in other major capsid proteins in viruses infecting all domains of life. And yeah, but still all these other phages and viruses, they have a double-stranded DNA genome, whereas so far Flip is the only one with a single-stranded DNA virus. The DNA molecule. So, and we have suggested this Flip to belong into fin lake viridae just a couple of years ago, since it doesn't resemble other known phages or viruses. So, Flip was originally isolated in 2010 in September during a laboratory course by students. So they were collecting phages and bacteria from the environment, and then just by accident we found this phage. And we've started the study it, and we have been always really interested in finding out the kind of what kind of phage bacterium interactions it has and what kind of ecology, etc. So we know that this phage, it's a unique isolate, but it has it can infect 3 different flavobacterial hosts in our collection. So the B330, which is marked in the bold text, that's its isolation host, but also it can infect other 2 hosts, which is interesting that this B114 has been isolated from very northern part of Lapland, for example. So it indicates that the hosts for this phage are available in different kind of environments. Also, another kind of metagenomic study have found that this major capsid protein sequence can be found globally from different metagenomes and from bacterial genomes. So this phage and phage type is more widespread than we know. But in my lab, so we have been studying how different kind of culture conditions, for example, temperature and oxygen level influence flip interaction with these different bacterial hosts. And this is, of course, important, as I mentioned in the beginning of the talk, that we have quite a wide differences in the seasonal conditions in Finland. So during the winter time, the lake is covered with the ice cover, and then in the summertime, the light is penetrating the surface waters, and we can have blooms of bacteria and algae. So what kind of roles do these kind of environmental factors play for flip infectivity in the laboratory conditions? But then we've also, in 2021, we started a large-scale study to sample the same place that the flip was originally isolated from, to see if we can re-isolate flip and what other things can we learn from flowobacteriophages. So these are the points I'm going to talk to you. So, these are some pictures of a flip infecting the 3 different flowobacteria hosts. So first of all, what we've learned that flip doesn't really infect flowobacteria in the liquid culture. So we can't propagate it efficiently using liquid. That's why we usually use plate lysates to propagate the phage. And this is probably because the flip might use part of the gliding motility system as a receptor. And this gliding motility system in flowobacteria is expressed when it's attached on surfaces. So it might be that these bacteria need to be producing some sort of biofilm, or at least be adhered on some surface for the flip phage to be able to infect them. And yes, so the also other factor that influences the flip infectivity in these 3 different bacterial hosts is the age of the bacterial culture. And this is quite rather interesting, and it happens in different ways in different hosts. So for example, with the B103 330, the isolation host of flip, we have to use very fresh bacteria culture. So we do the overnight culture, and then we renew it and have it in a shaker for a couple of hours. And then when we infect it with the flip or on agar plates, I don't know what happened here. How do I get back? From there, yeah, sorry. Yeah, so it needs to have fresh bacterial cells in order to produce very clear flux and efficient infection. Whereas in the other bacteria, the 114 phage, 114 flowobacterium, which is isolated from Lapland, there the flip prefers to infect the older cells. So it produces more efficiently the flux when the bacterial cells have been cultured just overnight and not diluted before the assay. Although in B114, usually these flux remain quite turbid. And we've also done some adhesion, adsorbance assays, which you can see in the lower part of the figure. And interestingly, for example, for B167, the adherence capacity to those bacterial cells are quite poor, even though the flip can infect this bacteria. And here again, we can see differences whether the bacterial cells are fresh or kind of older on the dynamics, how flip is adsorbing into those. So obviously, when we then think about the seasonal dynamics, and we want to know how does the culture temperature influence flip infections. So then we have done assays where we just do the double agar assay with the different bacterial hosts, and then we do the titrations on the plate and then incubate these plates in different temperatures. And what we can see, first of all, is that all these bacterial hosts, they are very secret tolerant, so they can grow in all of these temperatures from +4 until even to +33, which is a very rare event of having such water temperatures in Finland. But the most kind of even bacterial load can be detected when we culture the bacteria from 7 degrees until 25. But what happens then with the flip infectivity? So flip can produce plaques in all of these hosts, obviously also in the cooler temperatures. It most efficiently infects its own isolation host during all of these temperatures. And what is also interesting is that this B114 phage, since it's isolated from Lapland, it's more kind of tolerable to the colder temperatures. It actually is able to a bit resist on the infections in the most colder temperatures. But what we can see on the agar plates is that especially in these first 3 colder temperatures, which I've marked with the rectangle, the loosest area on the drops where we have the phage, they are like double the size of the loosest areas that when the bacteria are cultured in the warmer temperatures. So somehow this culture in the cold environment, it doesn't increase the titer of flip, but it makes it more efficient in cell lysis. And this is something that happens also in the anaerobic culture. So here's an example plate where flip has been drop titrated on a plate with B167 bacteria. And the plate has been kept 4 days in an anaerobic chamber. And during that time, the bacterial host doesn't grow. So it's just on a very stressful conditions. But then when we lift these plates on kind of normal conditions where the oxygen is available, we can start to see the bacterial growth. And here as well, the upper part of the figure is flip, and then the lower part, this is just a double-stranded tailed control phage that we have on the same plate. They've gone through the same conditions. So interestingly, in flip, again, this kind of loosest area is so much bigger than with the control phage. So it also benefits from the oxygen stress created for the host. Now, these all then kind of point to the direction that it's really the environmental conditions have a very strong effect on the life cycle of flip. So next we moved on to see, can we detect flip from Lake Jyväsjärvi when we do a year-round sampling? So what we did is that from February 2021 until January 22, so this year, we did at least once per month, we collected large water samples. And we did all sorts, we collected the bacterial communities on filters, and we also collected the viral fraction, and we've done sent those to metagenomic sequences to explore these questions a bit further. But what we also did, that we used these 3 different bacterial hosts to isolate new flavobacterial phages from each time point. And here you can see just the temperature regime throughout that sampling period. The squares are the water temperature, so it really goes from nearly 0 degrees until 25 or so during the year, and starts to drop again. So interestingly, we started to observe flavobacterial phages only at the very end of the summer. So now this figure, it just tells you the number of new phage isolates from these samplings per each bacterial host used during the period. So you can understand, so during the first 6 months or so, we got very little isolates, and we got a little bit of frustrated and desperate even, because we can't get any new phages. What's wrong? Do we have some problems with our own methods? But then as soon as the water temperatures started to drop here, we started to see flavobacterial phages. So now we don't know the abundance of the flavobacterial hosts in this data set until we have the genomic data available. But it has been shown in other studies, and this data has been collected from Sweden, from different lakes, that flavobacterial blooms are connected to the seasonal variation, and they usually occur during the summertime. And this quite nicely then reflects what we see in our own data, so that if flavobacteria start to bloom during the summer, then it's kind of obvious that then the phages will follow those dynamics. So just to present this in a table format, so we got something like 80 new phage isolates, and we did some PCR, and we targeted the PCR for the flip major capsid protein and Rowley circle replication protein. And indeed, we were able to isolate 2 new flip isolates using 2 different bacterial hosts from this data set. And then they were isolated in August. And what we did next was that we sequenced these new flip isolates, and we saw that these 2 new isolates that were isolated with different hosts, they were identical to each other. But when we compare the sequence with the original flip isolate, we can see that this really is a highly similar phage. It has been evolving more than 10 years in that Lake Jyväsjärvi. It's 96.6 identical sequence. We can see also some amino acid changes. They are kind of spread along the whole genome, but also some places are collecting more mutations than others. It has a bit different host range than flip, and we are currently trying to understand if it has different kind of life cycle dynamics. Just to end the talk, I'd like to show you that now we have 2 isolates of flip, but we also have another isolate that has been induced from Flavobacterium strain in the 1980s, collected in Wisconsin. This also has a single-stranded DNA and the lipid membrane. The genome is a little bit smaller, and this also can be found from genomes of different Flavobacteria. So it's true that these flip type phages are probably more widespread in the bacterial communities that we thought. So I think it's my time to end this meeting. I hope I convinced you that the single-stranded DNA phages are important for microbial communities, and the environmental factors play a great role in their life cycle dynamics. Okay, and thank you for your attention. I'm happy to take any questions.
Hi, thank you for the very interesting talk. I have to introduce. So I'm Dibyendu. I'm a postdoc at the University of Exeter. You talked about how these phages seem to have a different affinity for the host based on their ages. So are you talking about whether they are like in exponential phase versus stationary phage?
Yes.
Phase?
Yes.
And that was different in different bacterial strains.
Yes, that's correct.
So have you looked at that further? Looked into it that further? Do you know, like, is it based on like some expression of some? You also talked about like the biofilm components may be important. So
so we don't know the details of that yet, unfortunately. Yes.
Yeah. One more question was, you also talked about how when each of these bacteria, since they have a broader host range, have you looked at the expression, the efficiency of plating between the different hosts? So when you find the PFU titers are different when you extract them from different bacteria, but do you like do an efficiency of plating comparison with another strain to know if that's like a generalist or a specialist? Does that change when you pass it through a different host?
No, we haven't done that. No, but that's a good idea. Yeah. Thanks.
Yeah. All right. I think lastly, you talked about also, sorry, too many questions, that you find the phages more often when it is in the summer.
Well, can't say more often. It's been isolated 2 times. So yeah,
okay.
but yeah, in general, the flavobacterial phages, yes. Yeah.
So do you find it as the phage as a protein separately, or do you find it within the bacteria? I couldn't.
Oh no, these are kind of free particles isolated from the environment.
Okay.
Yeah.
Okay. Thank you.
I might have missed the point, but the bacteria, is there the source of these bacteria? Are they from fish or what? Or they're just free living bacteria?
Yeah, they're free living in the water, just isolated from the water bodies.
Okay, thanks very much.
Yeah.
My name is Al-Hassan Al-Rifai. I'm a PhD student at the University of Sheffield. So my question is about, you have talked about that you have done a liquid lysate that wasn't successful. That's why you moved to the plate lysate.
Yeah.
So I'm asking about like what MOI did you use in this experiment? Was it like high or low?
I think that we've tried all sorts of things.
Oh, right.
Yeah. And it's also kind of very typical for flavobacterial hosts not to be lysed in a liquid, not just these, but also others.
You mentioned, sorry, the life cycle of the bacteria. So it can grow anaerobically, or it can grow aerobically, or it can go grow at different temperatures, right? Is the life cycle of the temperature or the life cycle of the bacteria different in all these different conditions? And does the life cycle of the phage reflects the life cycle of the bacteria?
Yeah, so obviously the growth is very slow in very cool temperature or cold temperature, and when we also go into the very extreme to the warmer end, so the optimal is somewhere in the middle, and so we don't really know yet how it is reflected to the life cycle of FLIP, other than what we can do more kind of on the agar plates.
Do you think if you normalize to that, you will see that actually the growth rate of the phage is the same at different temperature or at different oxygen levels. And it's just because it's depending, it doesn't have a polymerase, you said, for example.
Yeah, that's a good question. Unfortunately, we don't have any data to answer that. But
Thank you.
Yeah.
There's a question in the online audience as well.
Okay, one quick question and final question is there.
There's one question in the
Yeah.
Hello. Okay. All right. Thank you. All right. So my name is Amos Mohoni. I'm with the International Livestock Research Institute in Kenya. I was just wondering, when you checked the survival of this phage in different temperatures, there was somewhere it seems like it doesn't reach as far as 37 degrees.
I'm sorry, I didn't really catch the last part of the question. What did you
All
say?
right. From the temperature ranges that you showed, it looks like the phage doesn't survive in 37 degrees Celsius, right? If I'm correct from what I saw.
It does survive.
It does.
It does, yes. I'm not.
Okay.
I'm sorry. I'm probably not.
No,
yeah.
maybe you could find the slide with the temperature.
Yeah.
Okay, maybe bring up that slide.
Yeah. So just going there.
This one.
Oh yeah. I was looking at 37.
So does the culture grow? Does the culture grow in this? What's happening? At
The bacteria culture. Yes, so it's really slow. So this has been recorded, as you can see, you have to culture the bacteria there for a week, or the plate keep it for a week on the incubator to see the actual growth. So it's slower there, yeah.
All right, okay, thank you.
Yeah.
I think Lotta, Riina has intrigued everybody by this initial student project that's obviously intrigued all them and intrigued us. Thank you very much.